Good day, everyone. On behalf of CHI's Global Web Symposia Series and our sponsor, Quanterix, I'd like to welcome you to Predicting Progression of Neurodegeneration via Blood Test, NFL Protein for Therapeutic Development in Huntington's Disease. My name is Elizabeth Lamb, and I'll be the host and the technical director for today's event. Now I'd like to introduce our presenters for today. First is Dr. Edward Wild, Ph.D., MRC Clinician Scientist, UCL Institute of Neurology. Our second presenter is Lauren Byrne, Master of Research, Research Assistant for HD-CSF, Huntington's Disease Center, UCL Institute of Neurology. Now at this time, I'd like to introduce Kevin Hrusovsky, the CEO of Quanterix, to make a few opening comments. Welcome, Kevin.
Thank you, Elizabeth, and thank everybody for attending. We're really excited to have both Ed and Lauren with us today, but we also have Andreas German with us. He's basically our neurologist here at Quanterix. Very excited to be looking at new ways in advancing the overall field of looking at neurodegeneration and brain health via the blood. We've actually continued to progress the company over the last several years, launching new products and continue to evolve that overall investment in the future. You know, basically, we are digitizing ELISAs and enabling a sensitivity that was somewhat unparalleled prior to the technology that we are unfolding. And we believe that there's going to be someday as many as 4,000 proteins. Our current menu is about 100 proteins, and we've had over 200 proteins now detected using our platforms. But we do believe that the long-term opportunity here is to see disease before symptoms, to detect undetectable abnormalities, and to truly look at the baseline of health and any movements from that baseline. And so redefining the overall level of detection limit is a lot of what we've been focused on, and finding in particularly neurology, because of the blood-brain barrier and many of the proteins being somewhat restricted from entering into the bloodstream, that the concentrations are extremely low. And so there's been, I think, about 45 publications now, third-party peer-reviewed, in the area of neurology that's allowed us to further advance the field of being able to see brain health and also the beginnings of being able to see neurodegenerative disease. This next slide is just an indication of all the various biomarkers across just about every field of medicine. We've started with neurology, but we've got a very strong focus on oncology, cardiology, and infectious disease. And many of these markers, particularly around inflammation, cut across most of those disease categories. And being able to see them at baseline is what makes our technology very unique. And so many of these markers can't be seen at baseline healthy levels, and some of them can't even be seen at disease levels. And we're able to see, as you can see on this slide, across the board, we're able to get to not only the disease level, but also the baseline healthy level. This next slide just shows some of the work that we've done on several of the critical protein markers and the level of sensitivity improvement that's been demonstrated many times by our customers and collaborators. We now have about 160 instruments installed throughout the world, and we are launching another platform, a benchtop platform, later this year. This next slide just illustrates that we have a lot of publications across all the various categories, and we also have many markers now within those categories. And again, neurology, 51 publications and 46 different markers now. And it's a very significant area of opportunity, particularly as you look at Alzheimer's and many of the really debilitating neurodegenerative diseases. Being able to see them very early, pre-symptomatic, before dementia, is a major focus of our company and many of our current collaborators. This next slide basically illustrates that we started off with the HD-1, which is a fully automated technology. We have 80 off-the-shelf assays, and it runs about 400 samples per shift. And we now are launching the SR-Plex, which not only looks at proteins, but can also do DNA, RNA, and microRNA, and get the same level of sensitivity as when you utilize PCR without using PCR, eliminating some of the complexity of using PCR as well as some of the error in the AT bias that's created with PCR. And we're also evolving to point-of-care solutions longer term, and we're really excited about that advance. So this final slide just shows all the companies now that are utilizing the technology, and we are really excited that on October 24th and 25th, we're going to be having a summit called Precision Health. It's going to be in Boston. We're going to have over 45 speakers over the 2-day period. Half the focus of the meeting is on neurology, and we have all of the leaders from around the world will be there to present their most latest information, but we also have equally exciting tracks in oncology and infectious disease. So we hope that all of you will be able to attend. We actually are waiving any kind of fee. Initially it was going to be a charged event, but we had so many sponsors join that we're now able to allow this to be a free event. So we're really excited for this opportunity to really move the fields of neurology, oncology, cardiology, and infectious disease. Now with that, I'd like to turn it over to Andreas, who will provide a few comments leading into the talks today. Thank you very much.
Thank you, Kevin. I think we'll get right to it as we discussed this morning. So I think Ed will start giving an overview of the Huntington space, need for biomarkers, and then Lauren will follow with specifics with regard to neurofilament and some published and unpublished studies, and then we'll open it up for 20 minutes Q&A. And again, for people, as Elizabeth alluded to, who have questions, there is a chat box. So, this is an interactive forum. This is an opportunity to ask what you always wanted to ask, but never dared to ask.
Hi, everyone. This is Ed Wild in London. Thank you very much, everyone, for the introduction. I'm going to give a general overview of Huntington's disease and what we think distinguishes Huntington's disease among the neurodegenerative diseases when it comes to the potential role for biomarkers and ways in which we might be able to make biomarker progress that is fueled by the particular characteristics of Huntington's disease. And then I will hand over to Lauren, who will give an overview of our latest findings to do with neurofilament light protein. As you've already seen, there's no video, but we thought we would provide a snapshot of what roughly the situation looks like. Today's mascot is Riley, so anything that has already gone wrong or will go wrong during the rest of the presentation is to be blamed on this delightful, innocent chihuahua. So this is Huntington's disease, and without video it's difficult to convey, but these are some stills from a video of a patient of mine called Kim, who's allowed me to share these images. And what you can see is the movement disorder that gives Huntington's disease its name. It used to be known as Huntington's chorea, from the Greek chorein, meaning a dance. We get the word choreography from the same root. And these are the jerky, uncontrollable, involuntary movements that are the most obvious feature of Huntington's disease. And they are disabling, but they're not the most disabling feature of HD. The movement disorder is more complex. Patients lose control of their voluntary movements, but even more disabling than that are cognitive decline, essentially a form of dementia, accompanied by behavioral and psychiatric problems. And so from very early on, the patients with Huntington's disease develop multifactorial disability in many domains, impairing their ability to work and function. And unfortunately, most of those symptoms are very progressive. Patients usually develop symptoms early in adult life and then progressively decline, but it's a disease that has a particularly long reach. Typically, patients will die 20 to 25 years after the symptoms begin. So patients are disabled for a very long time, and the collateral damage on other family members is huge. This is a particularly striking example of one of our patients. This is Brody, who was 10 at the time of this video, a good example of a patient with juvenile onset Huntington's disease. Her mother on the right was nursing 3 generations of her family at the time that this photograph was taken, highlighting the autosomal dominant inheritance of Huntington's disease. The cause of HD was identified in 1993 after a huge international collaborative effort. The gene is called HTT, and it encodes a protein called Huntington. The mutation is slightly special in that it is a CAG triplet repeat expansion, and each CAG encodes one glutamine in the Huntington protein, such that if you have too many CAGs, you end up producing a Huntington protein that contains too many glutamines. And in effect, if you have 36 or more CAGs, you produce the mutant Huntington protein, which is toxic to neurons. Below 36, and your protein is wild type, not named after me. The cellular pathology of HD is complex. The Huntington protein has many functions, and its mutated form produces many distinct and overlapping cellular dysfunctions. Each of these, for the optimist, represents a therapeutic target and the opportunity to intervene in a meaningful way. But of course, the most proximal pathology is the gene and the protein itself. So thanks to the gene being discovered, nearly 100 clinical trials have been run, and that represents just a fraction of the global scientific effort, which has included, of course, much basic and animal research. In the past 24 years, however, the results have not been particularly promising for HD patients so far. We have 2 drugs licensed by the FDA, tetrabenazine and deutetrabenazine, both of which control chorea but don't affect the progression of HD. So HD is currently impossible to prevent, slow down, or cure. But I think it's safe to say that the current generation of clinical trials are both more numerous than previous trials and also based on a much better understanding of HD pathology and are targeting HD in ways that are informed by our knowledge of the genetics and pathology of HD. One trial in particular stands out from this list. The list itself will be published in the next edition of the Journal of Huntington's Disease as part of a regular feature called Clinical Trials Corner. The trial that I think represents the state of the art in Huntington's disease therapeutics is the IONIS HD-TRX trial. This leverages the basic dogma of biology, namely that DNA is transcribed into RNA, which is then translated into protein. The mechanism of these ASO, or antisense oligonucleotide drugs, is that they bind to the free mRNA and result in its degradation, and as a result less of the protein is made. There's a review in the Lancet Neurology which came out yesterday by coincidence by myself and Sarah Tabrizi, which sets the scene for these and also tries to set out the landscape in this field. And as you can see, it's a surprisingly popular field. There are a number of different ways that we can try and engage with this whole pathway, from CRISPR to RNA interference and many options in between. The point is that we are in the age of Huntington modulation. We are engaging directly with the pathogenic agent of Huntington's disease, and these treatments are being given to patients, and we hope optimistically and realistically that we will start to see disease-modifying effects from these drugs which reduce the formation of the known pathogenic agent. So this brings us on to biomarkers, and biomarkers are a source of considerable debate, optimism, and much discussion. Let's say that. In Huntington's disease, I think the case is fairly convincing that biomarkers will be essential if we're going to achieve our mission. The known genetic cause of HD allows us to get a different view of the landscape. So if a neurodegenerative disease is like a river with parallel clinical pathways producing different features of the disease, and eventually, unfortunately, converging in most cases on death, then Huntington's disease really is a river where we get to see all the way back to the source. We know the source of our pathogenic river. It is the HD mutation, and we know that it produces this toxic protein. We'd like to treat the disease before symptoms ever begin. If we're going to do that, we're going to need to know whether the drugs we are giving are having a desirable biological effect long before the patient would be expected to develop symptoms. And from the outside, success and failure of a therapeutic agent look identical in a patient who hasn't got symptoms yet. So if we're trying to prevent the onset of HD, we will only succeed in this if we have really reliable biomarkers. And we can use trials in manifest Huntington's disease as a means of developing and proving those biomarkers, but at some point we're going to have to make a leap into preventative trials that are powered by biomarkers. And so I spend a lot of my time thinking about biomarkers, and it seems to me that it would be incredibly helpful to have biomarkers of the very early events in this pathogenic cascade. In other words, can we detect and quantify the causative agent? As soon as that protein is formed, though, as we've seen, multiple parallel pathogenic pathways form, they branch, they interact, they intertwine, and biomarkers of each of these pathways would also be helpful, so that we know if we're targeting a downstream mechanism, we can figure out whether we have successfully altered that mechanism, even though we'll also need biomarkers for the bigger picture as well. But it seems to me that a particularly useful kind of biomarker would be one that tells us about the process of neuronal degeneration and or neuronal death. In other words, if all of these pathogenic pathways converge upon damage and death of neurons, then a single biomarker that can report on that would be a very helpful adjunct to whatever upstream and downstream markers and other measures we end up using. So as an example of a very proximal marker, we began with a very intensive effort to quantify mutant Huntington itself, and this was a global collaborative effort, and the work of CHDI Foundation needs to be acknowledged as a major driver and contributor to this effort, as well as other international collaborators. This has been a really difficult job, and I think this encompasses a lot of the difficulty in this whole field. The proteins that we're dealing with are very tricky. They exist in very low concentrations. The biofluids that we're most interested in are very difficult to get hold of. And even if we can detect the protein, we need then to be able to measure changes. And in many cases, we're describing entire fields of human protein biology in the process of trying to produce a meaningful number that we can use to support our clinical trials. So the first generation of assay that successfully was used to quantify mutant Huntington in an accessible human biofluid was this TR-FRET assay, using these two antibodies, 2B7, which binds to the N-terminus of mutant Huntington, and MW1, which binds to the polyglutamine tract. And using this assay, which was much more sensitive than previous assays, we were able to show that mutant Huntington is detectable in pooled white blood cells from human blood from living patients, but not detectable in controls. And what's more, the level rose with progression of the disease. And this was great. We were hopeful that we might then be able to apply that assay to CSF to report on what was going on in the brain. Unfortunately, mutant Huntington has high levels in every tissue, so we're under no illusions. This is a peripheral marker of peripheral Huntington production. This is not reporting on what is happening in the nervous system, unless it is doing so by mirroring parallel pathologies. But what we really want to do is interrogate the nervous system directly and biochemically, and so CSF is the natural tissue for that. Unfortunately, this assay was not sensitive enough to do that. So we essentially waited for the next generation of assays to come along while honing our methodologies and putting together samples. Using the same antibody pair, we were able, using a single molecule counting assay, this one not on the Quanterix platform, but more of this later. And for the first time, we were in fact able to quantify mutant Huntington in human CSF. So in no controls was there any kind of mutant Huntington signal in CSF, but a signal was detectable in almost all the mutation carriers. And what we found is that as patients go from pre-manifest to manifest HD, this is in two separate cohorts, the level rises by about threefold. We showed other data to support the notion that this is probably mutant Huntington being released from neurons in the CNS. And this assay will, needless to say, be of great interest to any trial program which seeks to lower the level of mutant Huntington. And these assays, which are now several-fold, looking at different antibody pairs to interrogate different protein fractions and different combinations, mutant versus whole, total Huntington, they remain under intensive development, and hopefully we will see further progress soon. So this was published a couple of years ago, and I think is an example of a success story in biomarker quantification happening just in the nick of time to have an assay available to support this clinical trial program. So moving on now to neuronal death and therefore to inexorably to neurofilament, it is no doubt familiar to many of you as a component of the axonal cytoskeleton, and our interest in neurofilament clearly was inspired by the work of many other people who have done very, very solid hard work to develop the assays and also to then apply those assays to various neurological conditions. Looking at neuronal damage markers began with tau. We showed that the level of tau protein in CSF was associated with that of mutant Huntington rather closely, and also that it was expanded in HD mutation carriers. We then turned our attention to neurofilament, which had actually been reported in 2008 to be elevated quite consistently in CSF in Huntington's disease, but at this time no one had looked in plasma. And we previously showed in CSF that again the level of neurofilament was closely related to that of mutant Huntington, supporting this notion that these proteins are being released from the same cells. The Quanterix HD1 analyzer, ironically, had never been used for studying HD samples until we formed a collaboration with Henrik Zetterberg, who many of you know as being a biomarker guru, legend, and thoroughly good egg. He, along with Kai Bleno and others, co-developed the antibody combination and ported this assay onto the Simoa platform, where it is now, I must say, having no conflicts of interest with Quanterix, an extremely useful and reproducible piece of equipment that is forming an important part of our work now. There are many situations now in which neurofilament has been quantified in neurological disease. This is an interesting finding. After physical brain injury through instrumentation, the level of neurofilament in plasma quantified on the Simoa machine rises rapidly within a month of the injury, and then is beginning to fall 3 months from baseline, has returned back to normal 6 months from the time of injury. And that's really the kind of time frame that does, that would be viable if we can rapidly reduce the concentration of mutant Huntington through our Huntington lowering efforts. The other thing that I find quite striking is this publication from a trial of fingolimod in multiple sclerosis, where it was shown that patients randomized to fingolimod 12 months later had significantly lower levels of neurofilament. So I think this is the first pharmaceutical intervention that was associated with a reduction in serum neurofilament in a randomized clinical trial. And I think it gives us hope that this may be a useful marker of neurodegeneration, with caveats around blood-brain barrier and the different pathologies that we're dealing with. So the hope was that we would be able to apply this assay not to the river itself, but to the land adjacent to the river, in the hope of detecting whether there was contamination in the water without getting our hands wet. The analogy gets a bit tortuous at this point, but the point is that we wanted to try and measure neurofilament in blood in Huntington's disease. And with that, I'll hand over to Lauren, who did the bulk of the work on this project.
Thanks, Ed. So our work focused on a cohort called Track HD, which for anyone who isn't in the HD field, this was a 4-year longitudinal observational study, multinational, 4 sites across the world: London, Leiden, Paris and Vancouver, and recruited early HD and pre-manifest participants, including age and gender matched controls, usually family members or spouses. And all of these participants went through an intensive battery of different assessments, including 3T MRI, there was quantitative motor and cognitive assessments, and oculomotor, psychiatric assessments, and functional assessments. And also, as an ad hoc, blood sampling was done without too much idea of what they were going to use it for at the time. And as you can see, these papers were published from 2009 to 2013, and in 2017 we're getting something quite juicy. So the study design was very simple. We had blood samples from the baseline visit and mostly from 36-month follow-up, though a few of the participants we could only get from their 12-month or 24-month. And we wanted to look at how this single measurement of neurofilament related with disease stage at a current point in time. So just looking cross-sectionally at neurofilament levels and disease stage. So I think it's important to note here that the definition of pre-manifest and manifest in HD for anyone outside the field, this is clinically defined by the onset of motor or specific motor symptoms, and one of the kind of huge problems in clinical work in HD is that there are symptoms prodromal stage of disease with this, but no to classify that for clinical trials. And then both groups are further stratified into early and late pre-manifest. So early being subjects that could be up to 10 years, over 10 years, but from their predicted onset. So as you can see in control, so just to note here, the measurements of neurofilament are on a log scale. This was used to normalize the data. We have a few outliers in the controls, but when we looked over disease stage, you see a quite obvious increase with disease stage in Huntington's disease, which we were very happy to see. As you can see, each of the disease groups were significantly different from controls. And more importantly, these differences were significant between early and late pre-manifest and late and early HD manifest and early HD, which is extremely exciting. This is one of the first, definitely the first bioflow biomarker. This is one of the first bioflow biomarkers that showed anything this significant change or being able to significantly distinguish between early and late pre-manifest and manifest disease. We then wanted to look at the relationship with NFL and age. Ed mentioned before about the genetic cause of HD being a CAG repeat, and we see this relationship with CAG and age, and often these combinations are used in the predictions of HD. When we look at controls, there's pretty much a linear relationship with the age and neurofilament levels, but we want to look at different CAG lengths and their relationship with NFL. And as you see, with increasing CAG length, there's a more increased non-linear relationship with age. So people with higher CAG lengths, their NFL levels are increasing much more steeply and then leveling off than people with than lower CAGs, which when we take the points away creates a very pretty figure, which was in our paper, and we liked it so much. We made t-shirts. This is Nancy Wexler, who led or drove the collaborative effort to find the gene itself. So
I might just chip in here to say one of the things that was most striking about this was that this was really the first time that any fluid biomarker in HD had shown a relationship of this kind with the CAG repeat length, with every single CAG stacking up in order from lowest to highest, a very clear link to the dose-response genetic basis of HD. And I think that really argues very strongly for a very close link to the pathobiology of HD that we've really never seen before, even in CSF.
Yeah, thank you. So the next thing we wanted to look at how a single measurement of neurofilament at baseline could relate to disease progression HD. And one thing we really wanted to look at is the pre-manifest subjects and whether there was any relationship with baseline neurofilament levels and whether they progressed to manifest throughout the course of the study. So we split the individuals that pre-manifest at baseline into those that progressed to motor manifest and those who did not. And what we see is baseline levels of neurofilament were significantly higher in those that did progress to manifest during the course of the study. And this, after adjustment for age and CAG, remains significant, which is indicating that there is some independent predictive value that neurofilament has over and above the already known predictors of HD progression. This is just showing us the same thing in a different way, looking at the probability of those remaining pre-manifest, and we split the groups by the medium of baseline neurofilament levels. So those that were above the medium were significantly more likely to progress or to progress to HD or manifest HD. So next we want to look at the other measures of HD progression, and particularly brain atrophy, as we know in other diseases and in other neurodegenerative diseases, we're seeing a link with neuronal damage and with neurofilament. We correlated these with the various brain atrophy measures. So these are the atrophy rates, so change from baseline to follow-up with neurofilament at baseline. So we have caudate atrophy. So anyone who is not from the HD field, caudate, a part of the striatum, which is very specifically affected in Huntington's disease. And we looked at whole brain, grey matter and white matter, and ventricular expansion. And we see striking correlation with global brain atrophy in HD. So this is a single measurement of neurofilament at the start of the study, correlating with disease, with brain atrophy, or the rate of brain atrophy over the course of the study. And these again were significant after adjusting for the interactions of age and CAG. So again, coming in on the independent of the genetic dose of Huntington's disease, there's some independent interaction or something that neurofilament can tell us independently from what we already know from age and CAG. We also looked at some clinical scores and cognitive scores, and this single-digit modality test, which is a very common cognitive assessment for Huntington's patients, remained significant after adjustment for age and CAG. We also looked in a separate cohort, a smaller pilot study of CSF collection in Huntington's disease, and we looked at CSF levels versus plasma. So this is the first time anyone's correlated CSF levels and plasma neurofilament levels, and we see here there's a very strong correlation with CSF and plasma levels, suggesting to us that what we're actually measuring in plasma is an indication of the central system pathology, and that is the origin of the disease. Obviously, there's a lot more we need to do to test this, and it's something a big focus of my current work in my Ph.D. And a question we hope to ask a bit more in depthly. So in summary, we've seen that plasma increases with Huntington's disease progression, and there's a striking relationship with CAG repeat length and age. Ed mentioned about the genetic dose response related to HD, so which we find particularly exciting. And it also predicts onset, brain atrophy, and clinical progression. And we've seen that blood and CSF levels are closely correlated. So we like to look at this if going back to other biomarkers, and particularly brain imaging biomarkers and how they relate to neuronal pathology. So if you look at a brain volume, a one-off scan, MRI scan, it could be compared to an odometer, a single measure of how far you come, the distance traveled. So where a brain scan would be the distance of how far someone's come in the disease process. Then if you look at brain atrophy, so if you think of a speed camera which takes an image at one point and then further down the line and uses the distance traveled in a certain amount of time to calculate a speed or a rate. This is the same thing when you're measuring brain atrophy or rates of change, and you need 2 scans over, say, a course of a year. But ideally, if you want to be able to measure the rate in one measurement. So neurofilament, we've shown that a single measurement of neurofilament at baseline is representative of the rate of ongoing neuronal damage at that time, and this is what we're thinking neurofilament in Huntington's is representing. A lot more needs to be done. I know of a lot of, if you have a new car and you want to check the brakes are working, I would quickly go look at the speedometer to check this, and in the same respect in a clinical trial, if you want to see if a new drug is slowing the disease progression, I know what I want to use. If you're interested in these results and want to see in more depth the full analysis, we've published this data back in June in the Lancet Neurology paper, and you can always email us and get in touch after, and I'll pass on to Ed to talk about some further work.
Thanks, Lauren. So the needless to say, this is a pretty exciting result that I think came about as a result of the nature of Huntington's disease, giving us a genetically homogeneous cohort who all share the same base pathology, but with that spread of CAGs, giving us that really powerful insight into genetic dose-response relationships. And I think that combined with this very sensitive and accurate assay, plus all of the fastidious work that went into making the TRACK-HD study so consistent, was why we see such a really striking set of patterns here and a really remarkable predictive power of neurofilament. This is, however, just the beginning of investigating neurofilament as a biomarker in Huntington's disease. In order for it to be useful in the ways that we hope it will, considerable work needs to go into validating it and establishing its strengths and weaknesses as a potential biomarker so that we don't end up being misled. So careful work is now going into assembling as much information as we can from existing sample sets from humans, natural history and clinical trials, and also from looking at the numerous animal models of Huntington's disease, ranging from mice to pigs, and we call this informally the Influx HD Consortium. I say informally, it's formal enough that we made a logo for it. We don't really have results for that yet, except that in the near future, I'm sure that you will be seeing that some animal models of Huntington's disease do display what I would call a neurofilament phenotype, and others do not. I think this will be very important for us to know, so in order to help understand where the neurofilament's coming from and what it means, and also what animal models will be good for modeling what we might see when we treat human patients. Turning back to mutant Huntington, now that we have the Simoa machine, the HD-1 analyzer, we are trying to replicate the mutant Huntington assay using the same antibody pair onto it so that we can hopefully take advantage of what may well be a lower detection threshold and what will certainly be hopefully improvement in the scalability of this work. In the near future, we may be needing to test many samples in parallel very accurately, and hopefully having this assay available on the Quanterix platform will enable us to do that. I'm the global chief investigator of the HD Clarity study, which is the first ever multi-site, multinational CSF collection initiative, currently running in 4 nations, in startup in another 6, with more to follow. And this will be the biggest CSF plasma and clinical data collection ever in Huntington's disease. The samples will be made widely available, and you can find out more and apply to use the samples now. I think we have something like 130 samples already in the freezer. So go to hdclarity.net to find out more. And that, I think, is the overview that we wanted to give you. This has been a huge collaborative work, so thank you to all the people named and unnamed on this slide. And thank you very much for your attention.
Well, thank you. Quite intriguing. So we've time for questions. This is Andreas again. As we said before, if we don't run out of time, we have about 17 minutes left for all the questions. I see my email address penciled at the bottom. I'll forward this to Ed or share with Ed and Lauren, of course, and the rest as well, and we'll be more than happy to answer those. So one of the questions which came in, and I think this was before you went through some of the slides for NFL, it's kind of a broader question in 2 parts, so I'll do one part first. For the markers you show, does include wild type, mutant Huntington, and then neurofilament light chain. Have the data been analyzed, or is there sufficient power available for an analysis at the individual patient level in relationship to disease progression? That's part number 1 or part A.
Not really. The closest we've come, I think, is with neurofilament in the TRACK-HD plasma samples, where there was sufficient longitudinal data to be able to look at longitudinal change for the first time. This was only possible to do in a group-wise way, and we did see significant progression, significant increase in neurofilament in plasma on average in Huntington's disease, the clinical meaningfulness of that against progression in the clinical picture is much less clear. And I think what's neurofilament in particular, one of its strengths is that it doesn't need to be measured longitudinally in order to give us meaningful information. If we want to look at clinical progression or brain atrophy, we have to do longitudinal measurement, and of course that introduces error from having to measure something twice and subtract the difference. There's no getting around that. Neurofilament is a sort of cross-sectional measure of the longitudinal rate of progression of one element of Huntington's disease, and likely an indicator of neuronal damage. What this means on an individual patient level, I don't know, someone else has observed, I noticed in one of the comments that there is overlap between HD and control, and I think that there is, it's also known that there are other sources of variability in neurofilament. So, if you have a head injury or a stroke, in addition to having Huntington's disease, that could easily cause alterations in your neurofilament level, and therefore I think that's certainly one important reason why individual variations in neurofilament will need to be interpreted with great caution. The plus side, though, of course, is that unlike other neurodegenerative diseases, we don't really need markers of risk or diagnosis in Huntington's disease. We know who's going to get it because they have this very, essentially completely reliable genetic test. And so it's really the combination of a positive genetic test result with something like neurofilament that we can then use to identify the cohort and then characterize that cohort in terms of its risk of future progression. And I think that in the clinical setting, in the future, the way that these markers could come to be used is that, I think we'd be foolish to rely on any one biochemical marker, but neurofilament rising to a particular range that is beyond what one would expect for an equivalent control of that age, I think that would be something that we might perhaps use in order to, say, decide to start doing regular lumbar punctures on a person to quantify the mutant Huntington. So, perhaps to guide a transition from blood-based monitoring to CSF-based monitoring. And that's how I think, in the future, we may end up using some of these markers. We'll be using them in combination, and of course, also in combination with imaging and clinical assessments.
Okay. There's way more questions coming in. So from one of the slides had control data and HD patient data, and I have to recall, it was the CSF or blood, but there seem to be, as always, in these clinically defined diseases, no offense to the clinicians on the call looking to Ed and Lauren here, it's the definition of the control. So can you speak to this again? Do you recall the slide here?
Yeah, so the two different studies here. One is the 360-odd patient TRACK-HD cohort. The other is the HD-CSF study in London. And in both of those studies, the controls were very carefully selected. So they were recruited contemporaneously. They are, generally speaking, the partners or spouses or gene-negative siblings of the patients, and they are subjected to all of the same assessments in parallel. I think a big source of error in biomarker work in the past has been the use of non-contemporaneous controls, disease controls, CSF from people with headaches, collected a year earlier than all of the disease samples, and so on. Those experiments and projects were done through necessity, but in both of these work and in all our ongoing work, we recruit the controls contemporaneously, and we run it, and the use of partners and spouses essentially helps to match for environment, diet, levels of stress and anxiety, and many other things. So that's always been our control recruitment strategy.
Okay. So it's fair to say that these are healthy controls, not necessarily neurological disease controls, infectious and headache related.
Oh, absolutely, yeah. And they're quite carefully screened for the absence of other neurological diseases. Again, we're not interested in neurological differential diagnosis in HD because we already have a marker for that.
Yeah, exactly. Okay, very good. There were some outliers just in the follow-up on this in some of the slides with very high NFL levels, if I'm not mistaken. Have you ever looked back at the clinical records to see what the underlying rationale or reasoning could be?
We haven't, and that was by design. We know from the literature that there are many conditions in which neurofilament can can increase, and these samples were given with the expectation that clinical feedback would not be given, and so that's a sort of ethical issue. In other words, the samples were all analysed in a blinded way by an anonymous ID. We're given the basic clinical data that we present, but there's no option of going back. One can certainly speculate that those biologically plausible, very high levels of neurofilament might be an indicator that something's happening in the brains of those people. But neurofilament's not validated for clinical use, and therefore we would not use it for guiding clinical decision-making, even though we share the curiosity of the questioner as to what might be causing it in those individual cases.
Just to note as well, we performed all the analysis with and without the outliers on a side note, and we got the same result.
Yeah.
So before we come to, I think you alluded to some of the how do we use this as a toolbox in drug development. A more technical question is beautiful work, so congratulations again. Does plasma NFL correlate with leukocyte mutant Huntington levels? I don't know if we have data, if you've looked at this already.
Have we looked in? No, we haven't actually. So unfortunately, in the Track HD study, the plasma was collected and it was beautiful, but leukocytes were not isolated. So we don't have leukocytes from Track HD in which to look at in that large cohort. In our CSF project, the same is true. So we don't have the ability to look in leukocytes and in neurofilament. But I think we've probably gone one better by looking at CSF neurofilament and plasma neurofilament. We have actually in our HD-CSF now quantified plasma neurofilament and CSF mutant Huntington, and the results are still being analyzed, but essentially it looks like we do see strong relationships between those two, which is what you would expect because we already knew that the level of CSF neurofilament and CSF mutant Huntington were closely correlated. There's really, spoiler alert for completely unpublished data, it really looks like there's very little to distinguish between the predictive utility of CSF and plasma neurofilament. CSF might, now this is looking cross-sectionally and in a natural history study. What we don't yet know is if you successfully lower mutant Huntington, what the time course of any successive alterations of either CSF mutant Huntington, CSF neurofilament, or plasma neurofilament might be. We are on the brink of being able to answer those questions, but that experiment using the human model of Huntington's disease is still in progress.
Yep, and so I guess related to this in not particular order, this was one of the comments, and I think it's an important point, is this stationary measurement of cell damage occurring at the time of the measurement, or the question here was, and I don't know if we have data, you have data and analyze the data, is this a kinetic parameter? It's kind of related. Is this a measurement of turnover? So what do we know about the stability, I guess, because this has some implications as to use in drug development, I think. Do you have data? I guess as you said, you're just on the brink. We need to do this again, data. Is there any hint of how to answer that question?
We have some limited data on short-term stability over several weeks. Yeah, we've measured in the current HD-CSF study, we've had 15 people that have opted to do a repeat sampling of CSF and plasma between 4 and 8 weeks after the first sampling, and we get very high, I think ICC of like 0.99 or something. It's very stable over that period.
But I guess
from other diseses and from any, sorry.
Let's say you refer to the Ionis.
From what I've read about
the antisense study.
No, this
data in this cohort to say
we don't have any data yet.
Okay. But this is on.
No, that study's still ongoing, so there are no data at all available from that, and that will clearly be the crucial experiment to answer many of these questions at the level of both mutant Huntington and neurofilament. Of course, if nothing moves for either analyte, we won't necessarily be any further ahead. But if something moves, the time course of that and the sequence in which it happens and the relative proportions of those movements will be of, I think, huge interest and will also hopefully inform the use of neurofilament across neurodegenerative diseases. And I think this sort of, in some ways, validates what we've been hoping
because I was about
for
to
a
say,
while, which is that
from the data we will see in familial Alzheimer's disease with at least better estimated disease onset, we see increases pre-symptomatic to symptomatic within the 6 months to 12-month period, which I think is a very exciting or intriguing news with regard to clinical trials. If the same word holds true for Huntington's as well, you know, I think you have truly the biomarker of the year, as I tend to.
Right, exactly. And of course, with the data from acute brain injury, the data from contact sports, and the data from multiple sclerosis really do, I think, give us great optimism that we will be able to see meaningful changes within a time frame that would be feasible for exploratory work to try and bridge that chasm from early HD to pre-manifest HD, if we get a signal that that any of these approaches is working in manifest Huntington's.
We have a couple, I think 3 or 4 more questions for the last 5 minutes here. One was in the animal models you alluded to. There are obviously multiple with various facets of the disease. Are there any data available already? I know you didn't present any. Is it too early to tell, or?
It's too early to tell. I've already said as much as I possibly can, which is that we've analyzed multiple animal models. Some show a neurofilament phenotype. Others appear not to, but this is very preliminary.
Okay. But is an animal component part of your influx? I like the acronym.
Thank you. Yes, exactly. That is essentially the Noah's Ark of neurofilament in Huntington's disease. We're basically trying to gather to us samples from every animal model, natural history, and intervention study that we can in Huntington's disease, and in a priority order, try and figure out which animals show elevated neurofilament and at what stage of the disease, and what the effect of an intervention that was felt by some other means to be successful in that animal. And we can hopefully, sort of fill in an empty spreadsheet with yeses and nos about this animal has a phenotype, this animal responded in a predictable way to a successful intervention. And then I think, we're not saying these are good or bad animal models, but we're saying, if you want to look at this neurofilament biomarker and start to dissect where it's coming from in these diseases, figuring out which ones have the phenotype and which ones don't could be very informative.
Yep. So I mean, in that context now, I guess you will have a brain pathology component to this as well. I know in the human world you showed some of the atrophy data, but where does the NFL actually come from in the brain, striatum only in other brain regions? I don't know if we have an answer to that question.
No, I think what was fairly persuasive to me from the longitudinal imaging data was that the more of the brain a particular atrophy measure encompassed, the closer the correlation was with neurofilament. So there were significant associations with every atrophy measurement that's used in Huntington's disease, but the correlation was much closer for whole brain, it was close for grey matter, and it was close for white matter. It was less close for the caudate, and that's not surprising because the caudate's, a tiny 2% of the whole volume of the brain, maybe even less. So, it's clear that neurofilament light is not a region-specific marker. So if you're looking for a striatal marker, keep looking. This is I think a region-agnostic marker of the total amount of something, and what we call it is a matter for debate. It may or may not be contributed to by neuronal death. It is certainly probably a marker of what we could loosely describe as neurodegeneration. We're now looking much more closely at the voxel-wise imaging data. I think we have some nice findings there to suggest that there are independent areas where both white matter and grey matter are predicted by neurofilament.
So maybe 2 more final questions. One is a quick one, I hope. Have you looked at family-specific age of onset, CAG number, and NFL differences? I guess the emphasis here is on family-specific.
Oh, I see. So I suppose the question is sort of asking if a family has other genetic modifiers that cause them to have an unexpectedly early or late onset? Is that predicted by neurofilament?
I think it's just in familial AD they predict based on the parents, which in Huntington's we don't really use that because
right, yeah, exactly. So we don't look at parental age of onset traditionally in HD. That is something that's looked at in genetic marker studies, genetic modifier studies. In Huntington's, we tend to, because the CAG is such a strong predictor of onset, we tend to adjust and analyze for age and CAG rather than looking at correlations. But I think what we have shown is that if...
No, I think
sure, go on.
The future actually has become the present in a way, which I think is exciting, as we said before, enabled by Quanterix here, as Kevin has made his introduction. So I guess from a toolbox perspective, drug development, it's fast forward 6 or 12 months. I mean, I think we understand we cannot run clinical trials without it, basically, but how would we use the different markers? I think I'm thinking about patient stratification, target engagement. If you can talk, I mean, I think we're at the noon hour, but maybe we can have 1 more or 2 more minutes to talk about this, and then I think this was very informative, at least, and very exciting from my perspective.
Yeah, I think it will be a while before we see any of these biochemical markers being used as a primary or secondary endpoint in a clinical trial. I think they will be exploratory endpoints. And I think we've said all along, the lesson from other diseases like HIV and cancer is that you develop your biomarkers and your therapeutics in parallel. The key thing is getting a drug that works. And, I think really you have to go to battle using clinical endpoint in the first instance. In those studies, you can use, you design them in a way that you can analyze the biochemical, biofluid data in a meaningful way. And I think you then put together a portfolio, together with independent work in natural history cohorts, that these biomarkers are meaningful. And I think a combination of a marker of target engagement, in this case mutant Huntington for the Huntington lowering project, and a marker of neuronal damage, neurofilament, I think that is a very powerful pair of weapons for going to battle against this disease. The leap into pre-manifest disease will probably have to take place in sort of smallish experimental medicine design type trials where you give a small number of pre-manifest people in an unblinded way a dose of the Huntington lowering drug, and you measure the effect of these therapeutics. And I think that will then make a very strong case that we can start to move into much bigger cohort-based trials. And in the future, I think we will end up measuring some of these markers clinically. As soon as we have drugs that work, we will want to know, long before we give the drug, what the biomarker state of a particular individual is. And that's a lot of work. The work has already begun, is ongoing, and we and many others are working as hard as possible.
I just want to add on to that about, because kind of answering another question someone said about an overlap with controls and HD. There's definitely a lot of overlap with the pre-manifest groups, but it's important to note that the controls in our studies are matched to all of the HD gene carriers, and we see a strong linear relationship with age. If you're looking, what I've been anecdotally looking at, and some of our new data looking at some of the younger pre-manifest, when you're looking at people at that age and their neurofilament levels, there's a much bigger difference, which hopefully will be very important, as I say, for stratification of these clinical trials, and when you actually make those no-go decisions.
You have to model things appropriately with age taken into account.
We're 3 minutes over the hour, so I want to thank you again. Also want to thank Riley for being so quiet in the background. Thank you all the participants.
He really is here.
we believe you. Next time Riley will do 10 minutes of his presentation. He will do. This is Ed's dog, as we talked about this. So I think that stay tuned. We'll hopefully do this again. Again, thank you to all the participants. We will follow up with the recording of the meeting. You have the email address at the bottom of the screen. Please feel free to send us all the questions we couldn't answer within the time period prevented. And with this, thank you again and have a good day.